Hello everyone, and welcome to our online lecture series for Bio 1. You've made it! This is the last recorded lecture for our online Bio 1 class, where we're going to talk about our last organ system in order to cover up the last bit of vocabulary you need to have completed our ecological, cellular, and organismal part of biology. Should you continue on to Bio 2, you'll explore the idea of DNA structure, genetics, and biodiversity. And after that, you'll have completed the first year science majors biology course. So, hopefully you feel somewhat accomplished from all this. But let's get to it. Let's get this done. When we're talking about reproduction, as we've done in the past, we need to look at variation as to how different organisms perform the process of reproduction. There are two general blanket terms two general blanket ways in which reproduction occurs, either through some sort of asexual means or some sort of sexual reproduction. We're first going to talk about our asexual forms of reproduction. And what are the pros and cons, and what are the types of organisms that perform this, and what are the different ways in which you perform asexual reproduction. Asexual reproduction has one major drawback, that all the offspring are genetically identical. And this is really a drawback. Biodiversity, not just across species, but within a species, is what allows these organisms to adapt to different environments. And if we are maintaining very little biodiversity, that species is actually at higher risk of diseases, of environmental change, of going extinct more broadly. So it's very important overall to find some way to maintain biodiversity or generate new genetic diversity in a population, even if that species is reproducing asexually. These organisms that reproduce asexually are oftentimes producing clones of the parent, but not always. There are some exceptions to this. The major pros or benefits to asexual reproduction are that you're able to generate lots of offspring. Rather than invest in mating strategies, you don't need to develop all of the organs, all of the mating practices, all of the energy in making a way to sexually reproduce. These species instead focus on just putting all their energy into the offspring and making lots and lots of them. This is a huge benefit when we're talking about colonization. Asexual species always seem to thrive when a new environment opens up. Say a volcano creates a new island in the middle of the ocean. It's asexual species that will thrive on that island first, that will create a habitat, create food resources, particularly when we're talking about plants, that allow other species to come in and then thrive. So there is actually a place for both asexual and sexual reproduction in our biosphere. Both are necessary, and both do things better in different ways. As to our methods of asexual reproduction, we'll start by looking at these coral polyps. These are animals. Corals are not plants. Coral polyps reproduce via a process called fission. I guess I should identify this as coral. This coral is actually feeding through these little hairs that it's got on it. They look really cool and really colorful and beautiful. Fission is an asexual reproduction where the organism splits into two even pieces. By splitting into two even parts, these organisms are genetic clones, have probably not traveled very far, and never produce any gametes. Gametes are your sex cells. So there are asexual forms of reproduction where gametes are used. In this one, not so much. Now, another way to asexually reproduce is what this cartoon of this hydra is doing. Now, these are very small organisms, just a couple millimeters in size, but again, filter feeders in the water. These hydras reproduce by something called budding. In the process of budding, there's some sort of outgrowth that is designed to break off. And we actually see both plants and animals do this. There are animals that do this kind of budding where a smaller organism will be able to pinch off and then float away and land somewhere else. And then plants also create organs and limbs that are able to fall off quite readily, like our corpse flowers in the greenhouse. If something were to break off, it's able to form a new plant. And they oftentimes design these, like the mother of thousands. That's another really cool plant that has many, many little buds. Again, its name is fairly indicative of that, mother of thousands. It can have thousands of little buds coming off of it and starting new organisms. Again, in budding, these organisms are genetically clones. Our next type of reproduction is modeled by this sea star here. It's called fragmentation. Whereas budding is a purposeful outgrowth, fragmentation is an accidental tear. Now you would think that ripping the arm off of a sea star would cause it to die. But in this case, this is the old arm, and it is growing new components off of it. And in this way, through the process of fragmentation, a tear or a rip or some, like a boulder falling on the sea star in the ocean and crushing off one side can actually cause them to be two new individuals that, again, are genetic clones. All different forms of asexual reproduction, making the best of what the situation offers. There is one type of asexual reproduction that utilizes gametes. And this Komodo dragon which is a little baby, is actually produced by this form of asexual reproduction. It's called parthenogenesis. And in parthenogenesis, the egg that's made is never fertilized. But it is either, there's a couple different ways to do this, but it either doubles its genome or retains all of its genetic material, so that way this organism has a full complement of DNA. This never fertilized egg begins to develop, without fertilization and goes on to form a new individual. Now, some organisms are what are called facultative in their parthenogenesis, meaning that they can have sex or reproduce via parthenogenesis, like the Komodo dragon. In some cases, where these lizards will find themselves on a new island, and don't find a mate, the females are able to produce more females and increase in number. In cases where the mate is around, sex will occur, and voila, you've got a wider diverse population that can flex into a good colonial species or a well-established genetically diverse species. Parthenogenesis is also common in bees. And other insects, where rather than having a diploid individual, one gender is haploid. And of course, if you don't need a second set of DNA in order to be the particular gender, of course you're going to be able to develop via parthenogenesis. Interestingly, the haploid males are made in bees, but then in order to become a queen, there's actually a special diet. So there's a little bit of environmental and genetic triggers in order to establish your hierarchy in the caste system of bees, but parthenogenesis is underlying that mating system. We'll talk more about gender determination in a few. So let's revisit sexual reproduction as a means of comparing it to asexual reproduction. Some space here. So, with sexual reproduction, obviously gametes are involved. Sperm and egg, micro, mega gametes are used, and sex will occur. This produces genetic variation. And in so doing, you create a diversity for adaptation to select upon. You also hide deleterious mutations or genetic load. We'll talk more about that in a second. This is a sometimes event, depending on the mating scheme that you have. But it's important enough that we need to classify it as a general benefit of sexual reproduction versus asexual reproduction. And of course, sexual reproduction oftentimes usually uses different individuals of different genders. And in so doing, you have different genotypes that will able to mix and combine. Of course, there are variants to sexual reproduction, different ways in which organisms can do this. Some things are hermaphroditic. Or display hermaphroditism. And in hermaphroditism, you have one individual being two genders, or both genders. And in this case, some individuals are able to self-fertilize, resulting in some inbreeding depression, potentially. And this inbreeding depression can manifest both in plants and in animals. So don't think of it solely as an animal phenomenon, nor is it solely a human phenomenon. The reason why hermaphroditism has some benefit is that it allows you to mate with others when mates are available, or mate with yourself when colonizing. We oftentimes see hermaphroditism in plants. Plants with perfect flowers are said to be hermaphroditic. Perfect flowers have both the male pollen and the female ovary and egg. But not all flowers are perfect flowers. This Jack in the Pulpit is a flower of one gender. So, not all plants have these perfect flowers. Not all plants are hermaphroditic. We do see plants with gender. And their gender determination is not something that we're going to cover here today. Now, when an organism like a plant, oftentimes a plant, is hermaphroditic and is able to mate with itself, we refer to that as a selfing organism. Selfing does not produce genetic clones, but has very restricted genetic diversity and the other phenomenon that occurs via selfing is an increase in homozygosity. And this increase in homozygosity is what leads to inbreeding depression. And generally speaking, the way that this works is, let's go ahead and draw two quick chromosomes and say that there are 5 sites along this chromosome. 1, 2, 3, 4, and 5. And chromosome 1 is broken at 1, 3, and 4. And chromosome 2 is broken at 2, and 5, and 4. At their other sites, they work just fine. Pretend that these are genes, right? These are loci on the chromosome. They happen to have genes underlying them that are either broken or not broken. Now, in this individual, if this was the genotype for one individual, they would have at least one working copy of 1, at least one working copy of 2, at least one working copy of 3, maybe 4 doesn't matter, or 4 being broken is not that big of a deal, and they'd have at least one working copy of 5. So in this way, they're hiding their genetic load. But if this individual was to self, it would be much more likely that they would inherit the same exact copy of the chromosome. Now, this individual would only have a working copy of 2 and a working copy of 5. Sure, both of their copies work, but do you ever care if you have 2 working blenders when you only need 1? I hope the answer there is no. This individual would suffer from inbreeding depression as a result of the increase in homozygosity. You can see that this individual is much more heterozygous, meaning their genotype at one locus is different from the genotype at another, whereas this individual has the exact same genotype across its 2 different loci. Now, in humans, this inbreeding depression can cause phenotypic effects. Perhaps the best known example of inbreeding depression in humans occurred within European monarchies in the late Middle Ages. Look at some of the artwork surrounding the Habsburg family, a family that originated from southern Germany and went on to rule many different kingdoms, including Austria, Hungary, as the Holy Roman Emperors, different principalities in Germany, Spain at one point, and they had quite the distinct look about them. Notably, what's referred to as the Habsburg chin. Now, this phenotypic effect also had other physiological effects on these human beings. By the last reigning Habsburg, or true descendant Habsburg, from massive inbreeding, a Holy Roman Emperor in the late 1800s, this individual was having 30 seizures a day, I believe, if I remember that statistic correctly. They obviously were not functioning normally as a person. There were lots of different neurological, developmental, and physiological effects that impeded their ability to function. So, inbreeding, bad within humans. To counteract this inbreeding, a lot of organisms are not selfing, but instead outcrossing. And outcrossing organisms need two individuals of different genders. Because one is going to bring a mega gamete, or a large gamete, and a microgamete from the other, the small gamete. In human cases, we see egg and sperm. Outcrossing masks genetic load much more effectively. And results in more variable offspring. Not only can this mask genetic load, but in some cases, we actually see the phenomenon of heterosis, or what is termed hybrid vigor. Because in some cases, outcrossing can lead to more than the sum of their parents. For example, if one parent has a height of 1 and another parent has a height of 2, if they happen to be at different loci, let's say here and here, by putting something that adds some height here and adds some height here in the same individual, now that child may be not only 2 feet tall, but there might be a interplay between those two that would make that individual 2.2 feet tall. And that's the idea of heterosis, that there is more gained than just what genetics would suggest. So I oftentimes term it as the 1 plus 1 equals 3 in terms of genetics. But there are some cases of negative effects by mating from something that's too far away from you genetically. These are called outbreeding depression. Not something that we're going to dive a lot into, but outbreeding depression will oftentimes occur via something called Dobzhansky-Mueller effects, or DM effects. And in these cases, it can be, it's most often found that these are negative reactions to the immune system, essentially, where something is so distantly related that the immune system will identify your own organ as something that should not be there. These Dobzhansky-Mueller effects seem to occur both within humans and plants. And interestingly, they might have a role in us. One way in which this manifests is the fact that we have mated with other Homo species. We are in the genus Homo. We are Homo sapiens. But in the past, we've mated with Neanderthals. And one recent finding is that individuals who carry more Neanderthal ancestry seem to have a risk for higher autoimmune disorders, which again is in line with potentially that outbreeding depression. So, with the basis of our different mating types, meaning asexual and sexual reproduction, we now can get into the topic of sex determination. So here we have the image from Wikipedia that kind of gives us an idea of the different chromosomes that are responsible for determining sex in different organisms. And yes, sex is often a genetic factor, meaning that you have to have a very specific genotype in order to be one gender versus the other. The cross with a circle is female. The circle with an arrow is male. And when looking at humans, we know that we are XX or XY in our sex chromosome types. So we're oftentimes looking at our sex chromosomes when looking at sex determination factors. And these sex chromosomes will carry genes important to determining the different genders. For example, the Y chromosome carries testosterone, which is the male determining gene, and the female X chromosome will carry high abundances of estradiol and the other suite of estrogen-related hormones. Now, a human will have 44 autosomes, which are your not sex chromosomes, or your body chromosomes, and then your 2 sex chromosomes. In our situation, the homozygous genotype is female, and the heterozygous genotype is male. This differs in different organisms. The XY type is something that we see oftentimes in mammals, insects, and some plants. In birds, fish, and a number of insects, there's just so much diversity in insects, we have another mating system type called the ZW. And the ZW type is different because in the ZW type, it is the heterozygous female and the homozygous male. So just different. These actually, the ZW and XY sex determining systems actually have shared ancestry, so you can trace them back, and they seem to have evolved from similar systems, just with a different chromosome carrying the male versus female determining factors, which is quite interesting. We also have cases in bees where, or in many insects, where you can still make a viable offspring if you are missing a sex chromosome. And this is what we're seeing here in this fly species. Females are homozygous, males are heterozygous or haploid. You oftentimes see this kind of system present in organisms that may use drones or something of the sort, right? Where an unfertilized egg will develop into one type of gender, and that gender will be more frequently a worker of the group. Now, this is different than our last type of genetic determination factor here, called haplodiploidy. Here it is an amount of information that determines gender. And here we see most ants and bees and their drone factors here. Drones being those worker bees. In females will be homozygous and diploid. Males will be haploid. And they have no Y chromosome. Therefore, the only way to make a male is to have an unfertilized egg. A lot of diversity for these different mechanisms of gender determination via genetics. We also have some non-genetic ways to determine sex as well. For example, in crocodiles and other reptiles, we have environmental gender or sex determination. In this case, the temperature around the organism during its incubation will determine the sex of the offspring. For example, in turtles, we see that cold climates or colder weathers will skew towards males, and warmer weather will skew towards females. This type of gender or sex determination can be a little bit more problematic when we consider things like global climate change. It has the potential to skew the gender ratio in a species very strongly towards one type, either a whole bunch of males or a whole bunch of females, depending on the fluctuation in the environment. So there's a little bit of risk involved in this, particularly right now, but you should be aware that this is one way in which it is done. Another way of sex determination actually allows for sex swapping. And this is common in fish, like the clownfish. And this is my moment where I try to perhaps ruin some childhood memories regarding the movie Finding Nemo. Clownfish are able to switch genders, and in the event of a missing female, two males being present, one of the males would have switched to be a female. So in a biologically accurate retelling of Finding Nemo, it's highly likely that Nemo would have switched genders and had the opportunity to mate with his father. Sorry for that. But sex swapping is a thing in fish. Moving on from that potentially scarring event, I also want to talk about why X and Y or Y, Z, and W are, what's the history behind this chromosomal sex determination. And some of the coolest evidence we have is actually in this image here. This is the Y chromosome, and this is the X chromosome in this male in human chromosome spread. Now, what we've done is we've labeled a certain region of the chromosome that is homologous to the other, meaning that this region shares ancestry with this region. X and Y chromosomes are no longer able to recombine because they're so different. But there was a time way, way back in the day, before our species had sex or gender determining factors like this, where those chromosomes would have been similar enough to recombine. And it is the evolution of reproduction and the evolution of these chromosomes from shared ancestry that now they are so different that they're used to determine our genders. This is a very fascinating observation, and lots is going into this to study how these different sex-determining genetic factors share history and how they vary. But it is important to understand that reproduction, and in particular our genetic sex-determining factors, have evolved from common ancestry. Mating itself and reproduction itself has evolved as well, from unicellular organisms and early animals or primitive animals like earthworms that maybe don't have all of the same trappings of meiosis and reproduction that we do. We see different organisms develop specialized cells, like the gametes, then retain cells, like eggs within their body, and even innovate on the process of mitosis to arrive at meiosis. Once we got to our land vertebrates, we also see adaptations occurring in terms of sexual reproduction. Early land vertebrates still share a cloaca in common with our fish friends. The cloaca is an adaptation of the digestive tract to allow for reproduction. It is an orifice through which excretion, so urine, digestion, and reproduction occur. Birds still have a cloaca through which to reproduce and poop and pee. This, of course, differs from modern mammals, which have a uterus and a vagina on the female gender for reproduction. Now, the last topic that I've left some space for here talks about vocabulary surrounding the evolution of reproduction across the tree of life. These terms are oviparity, ovoviparity, and viviparity. And they define our systems of fertilization, incubation, and nutrition. And they have to do with eggs, the female gamete. For example, some types of fertilization are external, meaning that the egg and the sperm are released to the environment and allowed to meet and fuse. And this external fertilization is oftentimes aquatic, because obviously the sperm and the egg need to be able to move around, and water is much easier to move through. However, there's another type of fertilization option, and that is internal fertilization, meaning that fertilization is occurring within one of the two genders. There are many variants as to what happens to the fertilized egg afterwards, and that's what these three terms refer to. Oh, it's not oviparity, it's oviparity. Apologies for that misspelling there. In oviparity, we have incubation occurring outside the female's body, so the egg is laid, and the nutrition develops from the yolk. So oviparity is your classic bird laying an egg and it being incubated outside the mother. Let's go ahead and straighten these out. Incubation, nutrition, draw some grid lines here to separate our three different types. You also have a situation where the incubation will occur inside the mother, but nutrition is derived from the yolk. In this case, the eggs are retained in the female, and the egg yolk is feeding the young, but the mom is holding on to the egg up until birth. This occurs in many different types of some bony fish, including some sharks, lizards, and snakes. This is ovoviparity. In our last situation, incubation is going to occur inside the mother, and nutrition is also derived from the mom. Viviparity is what we refer to in most mammals. This is where mom is providing food for the young, oftentimes through a placenta. And so in this way, you have a catalog of the evolutionary changes in reproduction. Original reproduction was external, oftentimes aquatic, with the egg relying on the yolk, with the fertilized egg relying on the yolk for nutrition. Then fertilization in some lineages changed to be internal, with incubation still remaining on the outside with yolk-based nutrition. We call that oviparity. And then in some cases that egg was retained with mother, but the yolk still provide the nutrition, ovoviparity. And then we have mother providing nutrients in viviparity. And when mother provides the nutrients, it is retained within the mother itself. Whew. So that hopefully makes sense of these different vocabulary terms. Next up, we can take a human-centric view here as we go over the different organs involved in reproduction. Now, I'll highlight the things that I expect you all to be familiar with. Those being in our male reproductive anatomy, the scrotum that carries and supports and temperature regulates the testes themselves that produce sperm and our male hormone, testosterone, the seminal vesicles that contribute to semen production, the prostate gland that also contributes to semen production, the fluid through which the sperm will be expelled, and then the bulbourethral glands, which clean the urethra at ejaculation. Now, the reason why the scrotum and all of these, there are all these other glands that contribute to semen production, and all this temperature regulation happening with male reproductive anatomy, is that the proteins used in semen as well as in sperm denature easily. There is any kind of exposure to high heat will cause a breakdown in these proteins and will destroy them. So, therefore, male reproductive anatomy is largely focused on regulating temperature. Female reproductive anatomy has a few different organs, including the clitoris, the sensory organ, external structural organs, the mons pubis, labia majora, and labia minora, the greater vestibular gland, which secretes mucus, the breasts, which provide milk to the born offspring, so again, mother providing nutrition even after birth, the ovaries that carry and develop the eggs, of which there are 2 ovaries in humans, the oviducts or fallopian tubes that move the egg to the uterus itself that incubates the embryo, and the vagina, that is our common tube for intercourse, a birthing canal, as well as passing the menstrual flow. Now, interestingly, I want to talk about some of the cell types that govern or are common in these different organs. The oviducts themselves have cilia to help push along the egg after release. The uterus is lined with smooth muscle to help contractions in eventually expelling the fetus. And then the vagina is made of a lot of squamous epithelial cells, which allows it the features of lubrication and passing of menstrual flow. Now, while you may be familiar with some of these organs, hopefully you have a better understanding of what their exact purpose is through looking over these charts. I like to point out the developmental parallels between male and female, because at some point in your development, you had no gender. Before expression of these sex-determining hormones like testosterone and estradiol, we did not express a gender. Therefore, our development showed that different organs that are present in both male and female come from a same stem organ. The gonads are the stem organ for the hormone makers. In males, the gonads go on to form the testes, which there are two. In females, the gonads go on to form the ovaries. And they're unified by both making hormones. The urogenital folds in males make the penile skin. In females, they make the labia minora. The labioscrotal folds are perhaps the most telling in their name, as they go on to make the scrotum in males and the labia majora in females. Our last parallel that I'll ask you to keep track of is the genital tubercle. This structure will go on to form the penis in males and the clitoris in females. And this just goes to show you the effects of different hormones and the fact that, well, we are different genders, that developmentally we're not so different after all. There is there is shared history in our development that we all share when we were some few number of cells big, some, maybe a couple 100,000 cells big. There was no difference between the genders even. Kind of interesting to consider that. Now, we'll wrap up the lecture today by talking about a little bit about the ovarian cycles and phases, as well as gamete production in humans, and then end with our 5 developmental steps once fertilization does occur. So within humans, we have spermatogenesis, which is the process of meiosis in males. Spermatogenesis generates 4 gametes. These gametes are sperm with a long flagella that swims, as their task is to arrive at the female gamete. Now, the female gamete generates through oogenesis. And in oogenesis, rather than generating 4 viable eggs, 1 egg is generated. 1 gamete is made. So this is a very, very different outcome. 1 viable gamete versus 4 viable gametes. We'll explore this process a little bit later in Bio 2 when we talked about specifics of reproduction and revisit these ideas of polar bodies. And now for our ovarian cycles and phases. So here, I want to correlate hormonal changes with what is going on physiologically in the uterus, or our uterine cycles and phases. Now, in terms of the ovary, you have a follicular phase where the egg is growing, or the follicle is growing, a ovulation where the follicle is released, and then a corpus luteum, or a luteal phase, where things are degenerating and breaking down. You have this cycle repeating roughly every 24 to 28 days, I believe. Some variation around that. During the follicular phase, early on, you will see menstrual flow occurring. Then as we near ovulation, we have the proliferative phase. As the follicle is released and travels down the fallopian tubes, you have peak opportunity for reproduction. But then afterwards, there is a secretory phase that causes all of the uterine lining that has prepared for egg implantation to be released, to be recycled and regrown, so that way it is ready in case reproduction would occur the next cycle around. Now, these cycles are controlled hormonally, either through pituitary hormones or ovarian hormones. The pituitary hormones are follicle stimulating hormone and the luteal hormone, or LH. Now, both FSH and LH peak around ovulation. So it is the pituitary gland that's signaling for the timing of the release of the egg. The ovaries are producing two different hormones, estradiol and progesterone, that are on two different cycles. Estradiol will peak with FSH and LH, but then start to decline as you approach ovulation, and then it will rebound during the menstrual cycle, during the secretory phase, whereas progesterone will peak in the development of that lining and then will start to drop, inducing menstrual flow. So you can control the frequency, timing of the period through control of these different hormones. And it is, in fact, these hormones that are oftentimes what females will take as birth control. So very important to look into and understand what's going on with these different hormones in this very important cycle for both genders. Both males and females need to understand this process. Now, in terms of what happens if the follicle is released and it encounters a sperm, we can look at embryo development. There are 5 steps to embryo development. The first, the sperm will enter a region called the zona pellucida, which is a protective shell around the egg. It will penetrate the zona pellucida with lysosomes to eat its way into the egg, which is kind of crazy. Now, when the sperm has made its way in and the nuclei fuse, a zygote is formed, and the zygote is a diploid. As the nuclei fuse, the zona pellucida will prevent any other sperm from entering. This ensures that there will only be a fusion of one egg nucleus to one sperm nucleus. Now that you are a zygote, you will then begin performing mitosis. Your cell count will increase and increase and increase up until there is cleavage. And a hollow ball is made. That hollow ball being the blastula. Oh, with one L. Apologies. Whoa. What happened there? The blastula. Now, after the blastula is formed, mitosis will continue to occur, and you will develop more and more and more cells up until you develop layers in the process of gastrulation. Because that hollow ball will begin to pinch in and form a blastopore. That blastopore will be one of the two openings of your digestive system, either your mouth or your anus. And at this point, all animals look the same. Now, it will differ as to whether your mouth or anus forms first, as to whether you're a protostome or deuterostome. More on that in Bio 2. But when we are a zygote and a blastula on our way to gastrulation, we all look exactly the same, whether you are a fish, whether you are a lizard, whether you are a bird, or whether you are a dog. This is evidence of our shared ancestry. Once gastrulations occurred, we are now forming our 3 tissue layers. Our endoderm, the internal layer, the mesoderm, the middle layer, and the ectoderm, the external layer. And here you have some of the cell types made in each of these different layers. Obviously, during gastrulation, during gastrulation and the formation of our tissues, we now have a lot of differentiation. And it is in our similarity during gastrulation to organisms similar to us, like dogs and other primates, having more similarity in our gastrula, that allows us to infer even more about shared evolutionary history. The last step is organogenesis. And during organogenesis, you go ahead and make all of your internal organs. And this now separates all life, all animal life at least, into major different groups, and therefore we don't talk about it broadly, but rather specifically. And so hopefully this gives you some light on what's happening after fertilization, what's happening in female anatomy prior to fertilization, and the different cycles surrounding or involving the ovaries, egg and sperm formation, parallels in sexual reproductive anatomy, and then of course our different modes and benefits and minuses of sex determination and sexual mating systems. And lastly, thank you for taking part in this online biology course. Typically, in our face-to-face classes, I would now take the opportunity to shake everybody's hand and say thank you for a long semester of hard work. But hopefully this online greeting will do. I do genuinely appreciate all of the effort you put into learning all of these important biological topics and in becoming a better citizen of this planet. Biology will always help you understand your own body. And in understanding your own body, hopefully you can make good decisions for yourself and take those good decisions to being a good steward of our planet. Be good to one another, be good to yourselves, and be good to our environment. We all do have to live here. Let's make it as pleasant as possible. Thank you, and good luck on the final.